Assalamualaikum class. So here is the course content again, just to check what we have covered so far and what we are going to do next. So we have recently covered our series of lectures on bioenergetics and oxidative phosphorylation, in which we not only studied the concept of Gibbs free energy, but also we learned about glycolysis, TCA cycle, and electron transport chain. And now we move on to our next series of lectures, which is about carbohydrates. So let's get started. Carbohydrates, the most important and equally attractive thing in this world. Wow, just look at all these carbohydrates. I'm already drooling. When we think about carbohydrates, our thoughts tend to turn straight to bread. But carbs are found in many other foods too, such as whole grains, breakfast, cereals, rice, pasta, noodles, dairy, fruits, and starchy vegetables. They are also found in confectionery chips, cakes, pastries, soft drinks, sugar, and honey. One common misconception is that the benefits of carbohydrates provide can be found easily in other foods. But carbs are actually an essential nutrient, just like proteins and healthy fats. We need a certain amount of carbohydrates every day to sustain our energy level and fuel our body and brain. And research has also shown that grain consumers actually have a healthier diets and lifestyle patterns as compared to those who avoid core grain foods or core carbohydrates. Carbohydrates provide the body with glucose, which is converted to energy used to support bodily functions and physical activity. Carbohydrate consumed in the food yield around 3.87 kilocalories of energy per gram for simple sugars and around 3.5 to 4 kilocalories per gram of complex carbohydrate in most other foods. Relatively high levels of carbohydrates are associated with processed foods or refined foods made from plants including sweets, cookies, candies, table sugar, honey, soft drinks, breads, and crackers. Lower amount of carbohydrates are usually associated with unrefined foods, including beans, tubers, rice, and unrefined fruits. Animal-based foods generally have the lowest carbohydrate levels, although milk does contain a high proportion of lactose. Carbohydrate quality is also important. Some types of carbohydrates are rich foods and they are better than others. The healthiest source of carbohydrates are unprocessed or minimally processed whole grains, vegetables, fruits, and beans. They promote good health by delivering vitamins, minerals, fibers, and a host of important phytonutrients. Unhealthier sources of carbohydrate include white bread, pastries, sodas, and other highly processed or refined foods. These items contain easily digested carbohydrates that may contribute to weight gain, interfere with weight loss, and promote diabetes or heart diseases. Biochemically speaking, carbohydrates are the most abundant biomolecules on Earth. Each year, photosynthesis converts more than 100 billion metric tons of carbon dioxide and water into cellulose and other plant products. Certain carbohydrates such as sugar and starches are dietary staple in most parts of the world, and the oxidation of carbohydrate is the central energy yielding pathway in most not photosynthetic cells. Insoluble carbohydrate polymers serve as a structural and protective element in the cell walls of bacteria and plants and in the connective tissues of animals. Other carbohydrate polymers lubricate skeletal joints and participate in recognition and adhesion between the cells. More complex carbohydrate polymers covalently attach the proteins or lipid and act as signals that determine the intercellular location of metabolic fate of these hybrid molecules called glycoconjugates. In this series of lectures, you will learn about the major classes of carbohydrates and glycoconjugates, and I'm going to provide you with few examples of their structural and functional roles. Carbohydrates, as I told you, are polyhydroxyaldehydes or ketones or substances that yield such compounds on hydrolysis. On the right side of the slide, you can see the aldehydic and the ketonic group in a carbohydrate molecule. Many but not all the carbohydrates have the empirical formula of CH2ON. Some also contain nitrogen, phosphorus, or sulfur. Carbohydrates are essential macromolecules that are classified into three subclasses: monosaccharides, disaccharides, and polysaccharides. Carbohydrates can be represented by the formula which is written on the slide, which is CH2ON, where N is the number of carbons in the molecule. Therefore, the ratio of carbon to hydrogen to oxygen is 1:2:1 in a carbohydrate molecule. The origin of the term carbohydrates is based on its component, that is carbon, which is for carbo, and water, which is hydrate. Carbohydrates are classified into three subtypes, as I've told you: mono, di, and polysaccharides. Here are the three major classes of carbohydrates: monosaccharides, oligosaccharides, and polysaccharides. The word saccharide is derived from the Greek word saccharon, which means sugar. Monosaccharides or simple sugars consist of a single polyhydroxyaldehyde or a ketone unit, like I told you in the previous slide. The most abundant monosaccharide in the nature is the sixth carbon sugar D-glucose, which is also sometimes called as dextrose. Monosaccharides of more than 4 carbon tend to have a cyclic structure. Oligosaccharides consist of short chains of monosaccharide units or residues joined by characteristic linkage called the glycosidic bond. The most abundant are the disaccharides with 2 monosaccharide units. Typical is the cane sugar or sucrose, which consists of 6 carbon sugars, D-glucose and D-fructose. All common monosaccharides are disaccharides and they have names ending with the suffix "ose" or OSE. In cells, most oligosaccharides consist of 3 or more units. They do not occur as free entities, but they are joined together to non-sugar molecules, for example, lipids or proteins, and they form glycoconjugates. The polysaccharides are sugar polymers containing more than 20 or so monosaccharide units, and some have hundreds or thousands of units. Some polysaccharides, such as cellulose, are linear chains, and others, such as glycogen, are branched. But glycogen and cellulose consist of recurring units of D-glucose, but they differ in the type of glycosidic linkage and consequently have a strikingly different properties and biological roles to do. Now, when we study about carbohydrates, we need to know some important terms that will be helpful for a better understanding of the chemistry behind the carbohydrates. I have already explained most of these terms in our previous lectures, but still, just to refresh you, I'm going to explain each of these terms individually with reference to the carbohydrate to refresh your memory. So, first of all, we have isomers. Isomers are the compounds that have the same chemical formula but they have a different structure and they are called isomers. On the left side of the slide, you see a glyceraldehyde molecule and its mirror image. Now, this glyceraldehyde, which is the simplest carbohydrate, it exhibits properties of a chiral or optical isomer compound. And I'm sure you remember what a chiral carbon or a chiral center is, since I explained that in detail when we learned about the structure of amino acids. So this molecule, glyceraldehyde, it forms the basis of the designation of the isomers of all the carbohydrates. Glyceraldehyde can exist in two isomeric forms that are mirror image of each other, as you can see in this figure. The absolute configuration is defined by the molecule on the far left side as D-glyceraldehyde with the aldehyde group in the up direction. and the hydroxyl group must project on the right side of the molecule of the D isomer. Now, chemists have used this configuration of diglyceraldehyde to determine the optical isomer families of the rest of the carbohydrates. All the naturally occurring monosaccharides belong to the D optical family. Now, it is remarkable that the chemistry and enzyme of all the living cells can tell the differences between the geometry of one optical isomer over the other. Now, monosaccharides are assigned to the D family according to the projection of the hydroxyl group to the right on the chiral carbon that is farthest from the carbonyl or carbonyl group or the aldehyde group. Now, on the right side, you can see there is the structure of glucose and its isomers, and you see that the molecule which is numbered from carbon number 1 to carbon number 5 in the blue image 2. This is one carbon number 5, which is the aldehyde or the carbonyl group, and if this carbonyl carbon we have on the right side you see allose, altrose, glucose, mannose and galactose. They are all isomers of each other's and they have the same chemical formula of C6H12O6, but they have different arrangement of the hydrogen and hydroxyl group with the respect to the main skeleton of the carbon. So they are all isomers of each other. Second term that you need to know is apimers. Now you see here three structures of a sugar molecule. Now the two sugar molecules that differ only in the configuration around one carbon is called a apimer. Here you can see the D-glucose, D-mannose, which are different with each other only in the stereochemistry at carbon number 2. They are the apimers of each other. Similarly, they are also the apimers of the glucose and the galactose, which are different only at the position of carbon number 4. Then we have this term of enantiomer. As I've told you earlier, that an enantiomer is one of the two stereoisomers that are mirror images of each other and they are non-superimposable. Now, all the monosaccharides except the dihydroxyacetone contain one or more asymmetric carbon or the chiral carbon, or we can also call it the chiral center, and thus they occur in optically active isomeric form. Now, the simplest aldose, which is glyceraldehyde, contains one chiral center, the middle carbon atom, and therefore it has two different optical isomers or enantiomers, which are non-superimposable mirror images of each other. Now, by convention, one of these two forms is designated as D isomer and the other one is the L isomers or Leo rotatory isomers. Now, the two represent the three-dimensional sugar structure on paper. We normally use two types of formula. One is called the Fischer projection, which is shown here on the right side of the glyceraldehyde molecule, and on the left side on the other side you see a ball and stick model which is showing the actual configuration of the molecule, while on the right side you see the Fischer projection formula of glyceraldehyde, and on the lower right side you see the perspective formulas where a solid wedge shaped bond points towards the reader and the dashed wedges are actually pointing away from you. Now, the other term that you need to know is anomer and anomeric carbon. This is something new for you. Now, to understand anomers and anomeric carbon, you first need to understand the hemiacetal and hemiketal groups. Now, for simplicity, normally the structures of aldoses and ketoses, the straight chain sugars, they are represented as straight chain molecules, like you can see here. Now, in fact, the aqueous solutions of all the aldoses and all the monosaccharides with the 5 or more carbon atom in their backbone are actually present in ring-shaped structure, as a cyclic structure in which the carbonyl group has formed a covalent bond with the oxygen of a hydroxyl group along the chain. The formation of these ring structure is the result of a general reaction between alcohols and the aldehydes or the ketones to form derivatives called hemiesters or hemiketals, like you can see the reaction here.
Now, these hemiacetal and hemiketals, which contain an additional asymmetric carbon atom, and thus they exist in two stereomeric form. For example, we had D-glucose, which exists in a solution as a intramolecular hemiacetal, in which the free hydroxyl group at carbon number 5 has reacted with the aldehydic carbon number 1, rendering the later carbon asymmetric and producing two stereoisomers, which are designated as alpha and beta. Now, these six-membered ring compounds are called pyranoses because they resemble the six-membered ring, which is called pyran, which is like shown here. Now, the systematic names of the two forms of the D-glucose are D-glucose, D-glucopyranose, and the one is alpha and the other one is beta D-glucopyranose. Now, aldohexoses also exist in cyclic forms, having like five-member rings because they resemble the five-member ring compound furan, and therefore they are called as furanoses. Now here you see the six-membered pyran ring and the five-membered furan ring on the left side of the slide. Now isomeric forms of monosaccharide that differ only in the configuration about the hemiacetal or hemiketal carbon. As I told you, therefore they are called as anomers. Now the hemiacetal or the carbonyl carbon atom is the anomeric carbon, and these two forms of glucose, which is alpha and beta, they are interconvertible in aqueous solution by a process, and the process is called as mutarotation. That's a solution of D-glucose and a solution of alpha D-glucose and beta D-glucose will eventually form identical equilibrium mixture having identical optical properties. Now, this mixture consists of about one third of alpha and two-thirds of beta glucose and very small amounts of the linear and five-member rings or glucofuranoses. Ketohexoses are also present in the anomeric forms. Now, in these compounds, the hydroxyl group at carbon number 5 or carbon number 6, it reacts with the keto group at carbon number 2, forming a furanose or a pyranose ring containing hemi-ketal linkage. Now, the D-fructose readily forms the furanose ring, like you can see on the right side of the slide, and the more common anomer of this sugar is combined form or its derivative is the beta-furofructanose. So let's study the first class of sugars, which are the monosaccharides and the disaccharides. Monosaccharides, as the name indicates, mono means one, saccharide means sweet. These are simple sugars. In monosaccharides, the number of carbons usually ranges from 3 to 7. If the sugar has a aldehydic group, the functional group with the structure of RCHO, it is known as a aldose. And if it has a ketonic group, the functional group when the structure is carbon, which is linked with an oxygen and with the 2R group, it is known as a ketose, like you can see in the formula on the left side of the slide. Now, depending on the carbon or number of carbon in the sugar, they may also be known as trioses if they have 3 carbon, pentoses if they have 5 carbons, or hexoses if they have 6 carbon. Monosaccharides can exist as a linear change as well as a ring-shaped molecule in aqueous solution, and they are usually found in normally in ring forms. Now, simplest of the carbohydrate, the monosaccharides, they are either aldehyde or ketones, as I've told you earlier, with the two or more hydroxyl group. Now, the six carbon monosaccharide, for example, glucose and fructose, have five hydroxyl group. Many of the carbon atoms to which hydroxyl groups are attached are chiral centers, which will give rise to many sugars, stereoisomers, which are found in the nature. Now we begin with describing the families of the monosaccharides with backbones of 3 to 7 carbon, their structure of stereomeric forms, and the means of representing their three-dimensional structures on paper. Now, when discussed with the several chemical reactions of the carbonyl carbon of the monosaccharides, we are going to discuss that. One such reason, the addition of a hydroxyl group within the same molecule generates the cyclic forms of 5 or 6 carbon sugar, the form that predominates in aqueous solution and creates a new chiral center, adding to further stereochemical complexity these compounds. The nomenclature for unambiguously specifying the configuration about each carbon atom in the cyclic form and the means of presenting these structures on paper and therefore described in some details. Now, this information will be useful as we discuss the metabolism of monosaccharides in part 2 of the carbohydrates. Now, here, I'm going to introduce some important monosaccharide derivatives that we are going to encounter in later series of our lectures. Now, monosaccharides are colorless crystalline solids that are freely soluble in water, but they are insoluble in non-polar solvents. Most have sweet taste, and the backbone of most of the common monosaccharide molecules are unbranched carbon chains in which all the carbon atoms are linked by a single bond, a single covalent bond. In the open chain form, one of the carbon atom is double bonded to the oxygen, as you can see, and it is bound to an oxygen atom to form a carbonyl group. Each of the other carbon atom have a hydroxyl group. Now, if the carbonyl carbon is at the end of the carbon chain, then it will be called as an aldehyde group, and the monosaccharide is going to be an aldose. And if the carbonyl carbon is at the other position in a ketonic group, the monosaccharide is known as a ketose. The simplest monosaccharides are the 2 or 3 carbon trioses, for example, glyceraldehyde, which is a aldotriose, and a dihydroxyacetone, which is a ketotriose. All right, okay. Then monosaccharides with the 4 to 5, 6, and 7 carbon in their backbone are called respectively, such as tetrose, pentose, hexose, and heptose. Now, they are aldoses and ketoses of each of these chains lengths. Aldotetroses and keto tetroses, aldopentoses, ketopeptoses, and so on. Now, the hexoses, which include the aldohexose, D-glucose, and the ketohexose, D-fructose, they are the most common monosaccharides in the nature. The aldopentoses, for example, we have D-ribose and 2-deoxy-D-ribose are the components of the nucleic acids and nucleotides, as you all know. All the monosaccharides except the dihydroxyacetone, as I told you earlier, they all contain one or more asymmetric carbon or the chiral carbon, and therefore they are all optically active in nature. Now, the simplest aldose glyceraldehyde contains one carbon center, as you can see, and I have explained earlier on the left side, and therefore it has 2 optical isomeric forms or enantiomeric forms, which are D and L. For other biomolecules with chiral center, the absolute configuration of the sugar is known from the X-ray crystallography. Now, to represent the three-dimensional structure on the paper, as I told you, we use Fisher projection formulas, which I had explained in the previous slide. Now, in general, a molecule with n chiral centers can have at least 2 n stereoisomers. The stereoisomers of monosaccharides of each carbon chain length can be divided into 2 groups that differ in the configuration about the chiral center more distant from the carbonyl carbon. Those in which the configuration is at reference to the carbon is the same as to that of the diglyceraldehyde. These are the D isomers, or we also call them the dextroisomers, and those with the same configuration as L-glyceraldehyde, these are L isomers. So we can say that glyceraldehyde, whether it has D form or the L form, is actually the reference for making the isomers of all other sugars. Now, when the hydroxyl group of the reference carbon is on the right side in the projection formula, the sugar is the D isomer. When on the left side, it is the L isomer. Of the 16 possible aldohexoses, 8 are D forms and 8 are therefore L forms. The most of the hexoses of the living organisms are D isomers. The figure on the right side shows the structure of D stereoisomers of all the aldoses and ketoses having 3 to 6 carbon atoms. The carbon of a sugar are named beginning at the end of the chain nearest the carbonyl carbon. Now, each of the 8D aldohexoses, which differ in the stereochemistry at carbon number 2, carbon number 3, and carbon number 4, has its own name. And the name is D-glucose, D-galactose, D-mannose, and so on. The 4 and 5 carbon ketoses are designated by inserting a UL into the name of the corresponding aldose. For example, we have D-ribulose. It is the ketopentose corresponding to the aldopentose D-ribose. The ketohexoses are named otherwise. For example, fructose, which is from the Latin word fructus, meaning fruit, and it is rich in sugar, and sorbose from sorbus, the genus of the mountain ash, which has berries rich in related sugars and alcohol sorbitol. Now, the two sugars that differ only in the configuration around one carbon, as I told you, they are called epimers. Now, D-glucose and D-pannose, which differ only in the stereochemistry at carbon number 2, are epimers, and as are D-glucose and D-galactose, which differ at carbon number 4. Now, some sugars occur naturally in their L form. For example, we have L-arabinose and the L isomer of some sugar derivatives that are common compounds of glycoconjugates. Common monosaccharide sugars, for example, we have glucose, which has a formula of C6H12O6. It is the most common monosaccharide and an important source of energy. Now, during cellular respiration, energy is released from the glucose and the energy is used up to make adenosine triphosphate molecules, like we have studied that in the chapter of bioenergetics. Now, plants synthesize glucose using carbon dioxide and water, and glucose in turn is used for energy requirement of the plant. Galactose, which is an example of milk sugar, and fructose, which is found in the fruits, and other common are some other examples of the common monosaccharides. Although glucose, galactose, and fructose all have the same chemical formula, which is C6H12O6, they differ structurally and stereochemically. This makes them different molecules despite sharing the same atoms in the same proportion, and they are all isomers of each other, or we can call them isomeric monosaccharides. Glucose and galactose are aldoses, while fructose is a ketose, as you can see in this image, and from the formula and the presence of the ketonic group in the fructose molecule. Simplicity, all the formulas of or the structures of aldoses and ketoses were like represented as straight chain molecules. But in fact, in aqueous solutions, as I've told you earlier, aldoses or aldotrioses and all the monosaccharides with 5 or more carbon in their backbone, they occur in the form of predominantly as ring structures or cyclic rings, in which the carbonyl group has formed a covalent bond with the oxygen of the hydroxyl group along the chain. Now, the formation of these ring structures is the result of the general reaction between the alcohols and the aldehyde or the ketones to form derivatives, which are called hemiacetals and ketals, which I have already explained in the reaction, which is again written on the extreme left side of this slide. Now, these hemiacetals and hemiketals contain an additional asymmetric carbon, and thus, therefore, it exists in stereoisomeric form. For example, we have D-glucose that exists in the solution as an intramolecular hemiacetal, in which the free hydroxyl group at carbon number 5 has reacted with the aldehydic carbon number 1, and it has made its two forms, the alpha and beta glucopyranose. Now, aldohexoses also exist in a cyclic form, like I have explained earlier. However, the six-membered aldopyranose ring is much more stable than the aldofuranose ring, and it is the predominant form in aldohexose solution. Now, only aldoses have 5 or more carbon atoms and can form pyranose ring. Isomeric forms of monosaccharides that differ only in the configuration about the hemiacetal or ketal group They are called anomers, like I told you. Now, the hemiacetal or the carbonyl carbon atom is therefore called as the anomeric carbon. The alpha and beta anomers of D-glucose are interconvertible by the process, as I told you, was rotation. Thus, a solution of D-glucose will have the alpha beta form, which are being interconverted into each other, and they will have identical optical properties, right? So then about ketohexoses, they are also present in the anomeric form and they are also interconvertible. Now to represent the structure of D-fructose, which is in the form of a furanose ring, we use a type of formula like the Fisher projection that I told you. That is formula is known as Haworth perspective formula. Now, Haworth perspective formulas also are the way of representing these isomers on the paper. Look at the extreme right side of the slide, and you see the Haworth perspective formula. And these you see on the extreme right side, these are commonly used to show the stereochemistry of the ring forms of the monosaccharides. However, the sixth member ring pyranose ring is not planar, as Hewitt for perspective formula is suggesting, but it tends to be to assume either two types of conformation. If you look at the formula closely, you see it has somewhat chair-like conformation. Just to make you understand what is the difference between a conformation and configuration. Configuration is the arrangement of the parts of the elements in a particular form or a figure or a combination, while conformation is any of the spatial arrangement which the atoms in a molecule may adopt and freely convert between, especially by the rotation of about you can say a single individual bond. Now, look at part A of the Haworth perspective formula. These two conformation of the molecules are interconvertible without the breakage of the covalent bond, whereas the two configurations can be interconverted only by breaking a covalent bond. For example, in case of the alpha and beta configuration, the bonds involved, the rings involving the oxygen atom. Now, the specific three-dimensional conformation of the monosaccharide unit are important in determining the biological properties and function of some polysaccharides, as we will see in the later slides. The important thing about monosaccharide sugars are that they are reducing in nature. Monosaccharides can be oxidized by relatively mild oxidizing agents such as ferric or cupric ions. The carbonyl carbon is oxidized to a carboxylic group. Now, glucose and other sugars capable of producing ferric or cupric ions are called reducing sugars. This property is the basis of Filling reactions, which is a qualitative test for the presence of reducing sugar in any compound. By measuring the amount of oxidizing agent reduced by a solution of the sugar, it is possible to estimate the concentration of that sugar. For many years, this test was used to detect and measure the high levels of glucose in the blood and urine for the diagnosis of diabetes mellitus. Now, more sensitive methods of measuring the glucose level are available, but the main concept behind them was the use of employing an enzyme which is glucose oxidase for determining the sugar level within bloodstream.